We're going to move on to our panel discussion now, just so that we have as much time as possible. I'd also just like to introduce a new member of the panel, Professor Tao Dong, who's held the post of Professor of Immunology at the MRC Human Immunology Unit at Oxford since 2014. She's a Senior Fellow at University College at Oxford, and has also served a member of the UK Medical Research Council Infection and Immunity Board since between 2016 and 2020. She's the founding director of the Chinese Academy of Medical Sciences Joint International Centre for Translational Immunology here in Oxford. And she did her PhD with Andrew McMichael and Professor Sarah Rowland-Jones here in Oxford on qualitative changes in HIV-specific CTLs associated with HIV disease progression. And then in her postdoc, she continued to study immune responses to HIV, moving into influenza virus infection, which then led her on to start her own independent research group. And then in 2010, she became the head of the Human Antiviral and Anti-Cancer CTL Laboratory and a program leader at the MRC Human Immunology Unit at the university. So since the start of the pandemic, Tao's team has been working with colleagues in Oxford and in China, testing samples from SARS CoV-2 patients and trying to understand why some people with a COVID-19 infection are able to fight it off successfully, while others, of course, get very ill. Tao's team really believes in understanding the immune response to COVID-19 is the key to defeating it. So a very warm welcome to Tao and to all of our previous speakers who've already been introduced. I'd like to kick the discussion off really and ask you in turn what you think. So the first question really is, as viral immunologists, you've all rapidly moved from your existing area of expertise to working to characterize immune responses to SARS CoV-2. What has most surprised or intrigued you about the study of the virus? And we're going to start off by asking that question to Nina.
Okay, thank you very much.
Put you on the spot.
Yes, I mean, we moved really quickly to study SARS-CoV-2 infection, but actually we started off because we had a cohort here of having SARS-CoV-1 17 years ago. So this is actually how that we had studied before. Actually. So it's not, yeah, we are a research group that is usually working HPV infected patients, but we have done some work in SARS-1 infected patients. So it was not completely new area. I don't know what to say. I mean, compared to, of course, I work on COVID-19. For us, working on, or for me before, working more on HPV, working on the T cells and SARS-CoV-2 is, in a way, is nice because, to be honest, the T cells seem to be really present. There are a lot. They are to study them is, yeah, it's interesting for sure.
Great. Eui-Cheol, any other comments for you in regards to that?
Yes, actually, as the T-cell immunologist, the different virus is not the matter. I think that because just I can switch antigen, OLP or multimore, whatever. But one difference is before the COVID-19, like Nina or Antonio, or like my group, the T-cell immunologists, they were studying the chronic viral infection, like HIV, HBV, in my lab, HCV, for example, like that. But in practical way, there's a big difference between chronic virus and acute virus. So in terms of sampling, the sampling time point, for example, so for the acute study, acute viral infection, we have to sample frequently, for example. In the case of HIV, HBV, HCV, like once a year, for example. In that way, the big difference in practical way, but basically same, I think so.
Great, thank you. Tao, what about you? What do you think?
Oh,
put you on the spot again.
Actually, I agree. Same as Ewan, and I actually studied both chronic virus infection and HIV and flu acute virus infection, and in T cell response. Obviously, it's lots of common features and the differences, and also what's it's I think the COVID situation is really unfortunate situation, but at the same time, as a viral immunologist, I think it gave us chance to answer lots of questions we cannot answer in the like HIV chronic in setting or the flu setting as a general acute versus chronic virus infection. And these viruses have a really distinct feature. I think like it seems like get into the cells much easier, and you probably expecting to have a much more virus in it, and the disease pathogenesis is very different. It's about similarities and differences, but for flu, one of the frustration thing is it's very hard to get samples, especially the acute stage, because it's not routine practice. So COVID situation actually gave the immunologists a chance to be able to do at least large cohort type study and longitudinal to really have a big enough number to understand properly, although we're not there yet. But the samples collected and knowledge accumulated is something you would say for flu or HIV, it probably take 10 years. But now in 1 year, we get so many data and so many immunologists just jumping on it and work on it. It's actually, and we learn so much, but still we don't know the immunocorrelates and blah, blah, blah. But I think that we're getting there, and the knowledge and the tools generated has been amazing. So this is quite exciting. As a scientist, it's a really nice human model to study the T-cell immunity in the acute virus infection setting, and possibly a little because this long COVID and people with the immunocompromise is almost a bit like a chronic virus infection. So you can kind of, it's just a very good opportunity for us to understand the T-cell immunology and how the immune memory evolves and the contraction. So all these questions we had in probably all the T-cell immunologists had, but we can't really answer it, and the animal model probably can't really answer it fully. So I just think this is quite exciting opportunity for all of us.
Thank you. That's great. And Peng, I don't know if you want to add anything extra to that as well.
Yeah, just from like a practical issue. So first, when we look at the first Alice boat with PBMCs from COVID-19 patients, I was like surprised. Oh, the response is so strong. When after years you're working with HBV, HCV, and also cancer, you never see the strong response of this. And also when we isolate growing those T cells from COVID-19 patients, I just don't have time to look after them. And then after like 5 days, I found all the cells are growing like weeds. So this is, probably the studies like COVID infection as a primary infection is so different from the HBV, HCV chronic infections. Yeah.
Well, thank you. That's really helpful. Kind of interesting perspective. I guess some of the practical aspects, as well as more the immunology aspects of looking at some different diseases you're not used to. Eui-Cheol actually presented a question which I found particularly interesting, which was if, and we don't know the question here, but or the point, but if SARS-CoV-2 variants were totally evaded by vaccine-induced neutralising antibodies, so if the new variants were not, we didn't have protection with the current set of vaccines, would we be happy that vaccine-induced memory T cells still contributed sufficiently to our protection? So is a T cell response sufficient without the antibody cover? Because clearly it's been shown that the T cell response is very, very important with SARS-CoV-2. And I'm just wondering what each of you thought. Eui-Cheol, do you want to maybe expand on that? Yeah,
very good point. So upon your question, I can answer 2 points. The number one, I cannot remember, but it is supposed to be published in Nature Medicine, but I heard from some newspaper, but just I read the paper by MedArchive by someone, the Boston group maybe. So they once published that the hematologic malignant patient who was treated by rituximab, the anti-CD20 antibody. Sure, there's much higher mortality rate, but not 100%. The considerable proportion of this, the rituximab treated patient, they was spontaneously recovered from COVID-19. And they show that strong T cell response is correlated very nicely. It means that T cell response without any B cell, it still can confer the protection to the host. And second point, then the T cell response. So as you can expect, T cell response is much more durable to the mutation. Actually, in one Korean company now, they are developing the vaccine, but including not only spike protein, but also nuclear the protein. So now they are performing they just finished the clinical phase 1 trial. And the paper result was yesterday or the day before yesterday uploaded to the meta-archive, I think so. But in that study, I participated in that study. So we identified several epitopes from nuclear protein among vaccine. We found that like 6 or 8 epitopes, there's no mutation at all. So we just look through all the known mutation sequence. So it means that in this way, if we add one additional, in addition to spike, if we add M or any other non-structural protein, so we can make a second version 2 vaccine to more target to T cell. That's my idea. Yeah.
Mm-hmm. Yeah. Tao, what's your thoughts on that?
Yeah, first I want to echo Eui-Cheol's point about MP. Absolutely, this is what we see as well. We see, I think the immunodominant responses we observed are mostly actually focused on MP. Obviously, we didn't look at ORF1, and I realized ORF1 probably a lot of epitope, but MP is definitely a very, very good target. And another thing, I think what we see is in comparison with flu, COVID-induced the response we saw from the COVID patient is very broad. So that's very good news, because that is, unlike flu, it's very, very dominant. Like CD8, and if you look at the whole flu response, normally one individual probably only have 1 or 2 very dominant response. But for the SARS CoV-2, you probably have multiple. I wouldn't say hundreds, but probably quite a few, 5. I don't know. It depends on the individual. So this is for CD8. And so that means there's multiple target. This is probably the feature for the primary infection for flu, or although it's acute virus infection, but because it's a seasonal. So it's a kind of, the T cell receptor repertoire might be narrowed due to this frequent exposure. So I think it's quite hard for the virus to develop they probably develop one mutation, but it's probably quite hard to kind of escape all of the responses. But by saying that, it's probably, this is for the general case, but as individual cases, some people probably have a very focused response, and some are not. So those ones, there's still possibility for some people, like HIV, one single mutation in one epitope, like B2705 GAG responses is a very good example. Like HIV have very broad response, but this B27 GAG response, single mutation, is associated very strongly with the patient's rapid disease progression. So I think it's if you look at the population level, I wouldn't worry. But maybe at the individual level, some people probably should be worried. So I think that's why I think we do need to have this in-depth type of study to actually look further to really understand mechanisms. Another thing I think we should keep eye on the ones that in circulation, but not become a dominant strain yet. So there's in global database, we know this virus probably is not like flu or HIV that mutate that quick, but they still mutate. So there's accumulation of circulating virus, not necessarily driven by immune pressure, but it's there. So those are the ones maybe become dominant one day, we don't know. So those are the ones, especially the ones that are going up. And so I think then if we can test all these, keep eye on those mutations, and test them for their potential escape, then we're probably better prepared in the future.
Thank you, Tao.
Well,
Yeah,
that's what I think.
Yeah. No, thank you. And I mean, that's a really interesting point. And so Nina, you know, with your perspective of all the antibody measurements that you've been doing as well, and particularly in the T cell response in the severe disease where you saw kind of lower T cell responses, but still the kind of high levels of antibody. What's your perspective on that question about the importance of the T cell response versus antibodies?
I mean, I think always this idea of T cells versus antibody, I don't really like it. We do believe
No.
we have an immune system and we have T cells and B cells, and
They're
they
on the same
both
side.
play their role, exactly. So, and we can see, I mean, we studied now also antibody responses in, for example, in convalescence, but now also after vaccination. It's true that this mRNA vaccine is inducing an incredible high antibody titer, also for the neutralizing antibody. Some antibody titer we really see only otherwise in severe cases, which have probably seen longer antigen. But having said that, yes, I mean, of course, I think we are all happy that these vaccines are inducing also T cell response. You can see that they're inducing a broad T cell response to spike, so not only a single epitope. And yes, I mean, I do agree that we should probably, in the long run, think also to include other proteins into vaccine development for sure.
Yeah. Okay. Thank you. We don't have a huge amount of time, but I want to bring in some of the questions from our audience as well. So Marcia has asked, do we know if there are any SARS CoV-2 epitopes that are similar to self-peptide? So maybe cross-reactive, that may be inducing some responses. I mean, I don't know whether anybody's done any of those investigations or if anybody knows of any of those papers. Is that something that anybody wants to comment on? Unknown. Maybe unknown, yeah. So it's a very interesting point. I think particularly for maybe some of the other immune responses that we're not necessarily very clear of. The other question here from Gabrielle is quite interesting, which was, does anybody have an insight into people who are getting infected twice within a year, or presumably even more? My part of that is it with the same variant or maybe different variants? Is this something that's just due to the amount of infection, or some people don't develop a strong immunity, or something more specific with coronavirus? We obviously measure a very strong immune response to the virus in patients or those who are vaccinated. So why does an infection then occur? Peng, I don't know if you want to comment on that.
So I think we are studying some like reinfection patients, some T cell responses, like by collaborating with people from Sheffield. And one of the patients, immunocompromised patients, which has been through adoptive transplant. Yeah. So apparently those immunocompromised people, they are at high risk to get reinfected. But although we don't know, we don't sequence the viruses from first and second infection, but this is high risk for that group. Yeah.
Great, thank you. Eui-Cheol.
Yes, actually, but so another case is the related to the short longevity of the antibody, neutralizing antibody. So according to the several publications, I think that the compared to T cell, the neutralizing antibody have a shorter longevity, So I remember that the half-life is like the 5 or 6 months. So due to this, the disappearance of the antibody, maybe they get reinfected. So in that aspect also, I want to emphasize the T cell to alleviate to reduce the disease severity. Yeah.
No, but I mean, I think what is important is that we see a decline also, I mean, to be fair, also in T cells over time, even so it's less. I mean, I think the decline is really at the beginning and then it's sort of plateauing. And this is just that we develop then a memory response. And this is exactly occurring for T cells as for B cells. Yeah, the antibody titers might go down, but there will be persisting memory B cells and memory T cells, which then probably will not completely prevent reinfection. I guess we will all see a time when actually reinfection occurs, but what is important is that people don't get sick, right, or severely sick. If you then have a memory response, you get reinfected, but due to your memory T and B cells, clear the virus quickly without maybe even developing symptoms or just common cold symptoms. I think that's actually the goal of all these vaccination strategies we are doing.
Well, thank you very much, Nina. Thank you to all of our panelists. I really wish we had all afternoon or morning, wherever you are in the world, to keep on chatting. It's exciting, but we've got another panel discussion a little bit later, so this isn't the end of it. But I'd just like to put your hands together. You can click on the applause, but just want to thank all of our speakers, all of our panelists, for their contribution today and for presenting their data and for the chance to engage in discussion. There are lots of, so let's do a big round of applause. Thank you very much.